Yeah.
Let me see. Started. All right. Good evening, everyone, and welcome to our March 2024 Open Mic Night. This evening, we're going to be getting down and dirty with your soil biology with Lisa Blazure from the Stroud Water Research Center. We'll be hearing from her in just a moment. We also are joined tonight by Doug Adams with New Brooklyn Farms, who is working on a new initiative that may be of interest to folks. We'll hear from him. And also, I just wanted to briefly share with you some of our upcoming programs. So last month we did integrated weed management with Dr. Cerruti Hooks. This month, obviously, tonight we have soil biology with Lisa. I wanted to mention that Chesapeake Bay Foundation and NRCS, Colleen and I and myself from our office will be involved in a high tunnel system workshop at Claggett Farm. I think Isabella is going to be posting that on the Google group pretty soon, so look out for that. And then on April 16th, we have a cover crop workshop for small and urban farms at Edgewood Farms. That's the farm of Steve Darcy, our executive director. And we'll also have guest speakers from NRCS as well as the Maryland Department of Agriculture, talk about the financial incentive programs to help you pay for cover crop. And we're just going to have a nice, robust discussion. We're hoping maybe either late May, early June to meet on a farm and talk about water irrigation management. We have a few potential speakers for that, so stay tuned as we get closer. We'll narrow something down. Still looking for speakers for September and October. If folks have ideas, let us know. We also are taking some of the topics that you relayed at last month's open mic night and looking for some speakers. And then I also wanted to mention that Dr. Cerruti Hooks is going to bookend our open mic night schedule for this year with ecological pest management. So we're going to have him in November. And that is it for now. With that, I'm going to go ahead and kick it over to Doug, unless anybody else has any other brief announcements that they'd like to share.
Kim, quick question
Yeah.
about the cover crop event on the 16th. What time?
Yes.
What time is that? Is that at the same time that open mic normally happens, or is it earlier?
That is going to be a little bit earlier. I think it might be, let me just check my schedule really quickly. And I just realized I don't think I have my slides up for you all. I apologize. Let's see, that one is, it looks like it's 1 to 4 on my calendar.
Thank you. Awesome. Thank you.
No problem. All right. So with that, I'm going to go ahead and kick it over to Doug.
Thank you, Kim. All right. So hello, everyone. Quick introduction again for those who jumped on. My name is Doug Adams, owner and founder of New Brooklyn Farms, an urban farm and green event space located in Mount Rainier, Maryland. I have worked closely with Kim since I started in 2016. Crazy to say that. Yeah, come a long way, done a lot of awesome programming. I've operated a farm in residence program from my first season. Yeah, just learned a lot about, this game of urban farming and how some of the challenges really. So I think last year started in November, I was selected as a participant in a business accelerator program offered by the American Heart Association called Empowered to Serve. I was one of 5 farmers in Maryland to be selected for that, and went into it thinking that it was just going to equip me with some tools to kind of continue doing what I was doing already from a business perspective. It ended up kind of spurring a new idea and almost like an ancillary business from New Brooklyn Farms. So pretty much myself and Andrew Suggs, who's also on the call today, we're working together on a tech solution that is geared towards urban farmers and small farmers as well. An app-based solution that looks to accomplish 2 things. Number 1 is to measure and almost like really spur community engagement with urban farms. And then number 2, to kind of streamline a lot of the administrative data capture and just really impact reporting functions that a lot of urban farms need to thrive. I think that most of us know that a lot of urban farms and small farms tend to be nonprofit organizations. A lot of us struggle with high costs of doing business, labor, insufficient revenue from food sales, needing to supplement that. And yeah, we're just looking to really create an app that helps aggregate and measure all of the true impact, the dynamic impact really that urban farms drive for for their communities they serve from a health perspective. So with that, I pitched this idea at the finale of the Business Accelerator program and on stage and really in its infancy. And I'm continuing to work on it. Right now, we're in the market research and customer discovery phase. And my ask for the group tonight is Andrew and I have put together a survey to kind of just assess the needs of urban farmers and folks who are both operators and in the professionals in the space to kind of just share your thoughts on what some of the biggest challenges are, what they look like in your use case. And yeah, we put together a fairly brief survey that I'm going to drop the link to in the chat. But I just wanted to really just share that update with you all from the New Brooklyn Farm side. I'm still going to be, doing business as usual this season. Actually looking for a new farm in residence right now. I'll drop a link for that information as well in the chat. But really just that's really all I had to share. And then wanted to take maybe, Kim, if we had like 2 minutes for me to ask like one of these questions and just get some live feedback. Yeah, okay, cool. All right, so let me just first drop the link in the chat. Okay, it's there. And the main question I wanted to ask the group, and you can respond like in the meeting chat with your what you think is the top 3, but we were trying to grasp from your perspective of your urban farming operation or in your experience of what you've come across from urban farm operators, what are the 3 biggest business and or operational challenges that your farm faces? I'm just really I'm gonna read them off, but I'll also just drop them in the chat. And feel free to just just write back and let me know what for you is the biggest challenge. Listing them out, and so I'll read them all too. Fundraising expertise, business management, land access or land security, insufficient revenue from crop sales, inconsistent revenue from crop sales, high operational costs, social determinant of health issues that customers face, infrastructure maintenance costs, high labor costs, or lack of access to markets to sell product. So I just dropped that in the chat also. And if you want to just maybe we'll give it like 1 minute. If folks feel that they want to write back and give us some feedback, we would really appreciate that. So yeah, that's just pretty much like the biggest question of all the questions in the survey. But I did drop the link in the survey. If you have 10 minutes or so to run through that survey and complete it, it will really mean a lot to me. So Andrew, did I miss anything?
Yeah, I'll chime in. I mean, we're really here to serve you all. From a technological standpoint, a operator efficiency standpoint, as Doug has educated me on the industry of urban farming and just scouring the market to see what's out there that's solving some of the things that he mentioned. We're not seeing a lot, and so having a potential solution that's built from someone that's in the industry is the purpose of why we're trying to do this customer research, because from an entrepreneurial standpoint, nothing pains me most as a kind of successful, so to speak, entrepreneur than people building things that the market doesn't really need or want. And so with you all help kind of corroborating some of these 8 things that we think are like challenges based on the research and conversations that we're having, would be, you know, immensely helpful. So that's really it. We don't want to belabor. We can give you all time to complete the survey, but you know, that's really the biggest question that we would like to just get some initial feedback on. Like, what are the biggest challenges that you all have as operators, owners of your farms?
For sure. All right. Yeah. That's really it. Really grateful.
Yeah, let's see if we can. You know, I don't know if we have a few more minutes, but just see if we can get some response. I see we have some things in the chat. If anyone feels bold to elaborate, would love to foster, you know, just a couple minutes of dialogue.
We have maybe like a minute or two because I want to make sure we get to Lisa's presentation this evening as well. And I know we're starting a little bit late, but if folks want to, you know, mention a thing or two, maybe have a minute. Otherwise, maybe you all can follow up with, you know, Andrew and Doug.
Yeah, that's fine. Yeah. Thanks again, Kim. Really appreciate the time.
Absolutely.
Making room for us. But yeah, link is in the chat, and thanks again, everyone, for who's responding in the chat. Thank you.
And if anyone wants to share their contact information with Doug and Andrew for further follow-up, that would be great. And if there's enough folks that want to have like a separate, you know, half-hour conversation, I'm happy to, you know, loan our Zoom link if you guys wanted to pop in for something like that. All right. And also for folks who just hopped on, we're kind of done with farmer introductions, but if you wanted to introduce yourself in the chat box, please feel free to do so. Getting ready to introduce our main speaker for tonight. Again, Doug and Andrew, thank you so much for hopping on, and really excited about this app, and great job also at that business accelerator. That was really exciting to have a couple of Prince George's County folks involved in that. So this is kind of serendipitous. Lisa and I were on a planning committee for the Climate Smart Commodities Forum that we were planning with a couple of other stakeholders for December. And simultaneously, Michelle Coleman, thank you, Michelle, happened to be in a session with Lisa at Future Harvest and came to me and says, oh, there's this great lady, you know, Lisa Blazure, who gave this great talk at Future Harvest on soil biology. It's like, oh, Lisa. And so it just kind of was fun because I had just met, you know, Lisa, well, probably what we were planning that conference for a minute. So I probably met Lisa this spring before, but finally met Lisa in person in December. So that was fun that Michelle then, you know, popped up in January. So thank you, Michelle, and thank you, Lisa, for helping to make this happen. So I'm going to read Lisa's bio and I'm going to turn it over to her. Lisa is an experienced conservation professional who advocates for improving soil health to achieve water quality goals, grow nutrient-dense foods, and enhance farm profitability. She works for Stroud Center. Oops, I just lost my, hold on one second. I mean, you all don't need to hold on. I'm talking to myself
did
here.
I mess
My
you
screen
up
just
there? With...
No, it's fine. I just, you know, it went full screen. I was like, whoops, there went my intro. It's all good. She works for the Stroud Water Research Center and serves as the coordinator for the Pennsylvania Soil Health Coalition to foster collaboration among numerous stakeholders involved in soil health education and research. Lisa networks with a broad range of colleagues to advance soil health initiatives. Prior to joining Stroud Center, Lisa was the agricultural technician for the Clinton County Conservation District, yay, conservation districts, and worked in the Chesapeake Bay and nutrient management programs. She currently serves on the Pennsylvania No-Till Alliance Board and the Pennsylvania Sustainable Agriculture Board. So anyway, we are so excited to have Lisa with us today. We appreciate you crossing state lines figuratively to come talk to us in Maryland, and specifically Prince George's County. Yeah. So without further ado, go ahead, Lisa. I'm going to go ahead and spotlight you.
So easy to cross state lines with technology nowadays. So yeah, as Kim and Michelle mentioned, the stars just aligned for me to be able to share a topic that I'm really passionate about, and that's soil biology, right? And that's a real common foundation to any type of agriculture that we're working with. So a little bit about the Stroud Center. We're based down in Southeast Pennsylvania in Chester County in mushroom country. We've been around for more than 50 years, and we're primarily a freshwater research organization, streams and rivers. But we also have an education department and a watershed restoration department. And it may seem a little odd to have this water quality organization have someone like me on staff working with farmers and agriculture and talking about soil health. And it's because so many of our streams flow through these agricultural areas, right? And if we want healthy stream systems, we need to have healthy watersheds that are draining into those streams, right? So when it rains, the water in the stream is going to run clear, like this picture, the stream coming in on the left, and not running muddy brown and carrying away all that precious topsoil layer. So most of my education that I do is with kind of larger scale farmers, a lot of row crop, some confined animal operation. But when we're talking about soil health and really these core principles with building healthy soils, it works on any scale of agriculture, right? 1,000-acre farm all the way down to backyard gardening. So I don't have a farm myself. This is my little footprint in the world where I get to kind of influence and manage and grow my own crops. So I've got two different pictures here, but I did move about 1.5 years ago. I live in central Pennsylvania. So my old garden at the old house, I was kind of down in a hollow. I really didn't get any direct sunlight till almost noon. So I had at max maybe 7 hours of direct sunlight. And it really took me a lot of years to figure out what crops grew well in my soils. And those cool season crops did well for me. But you see, I've got 2 of those plastic composters. So I'm try to reuse and compost all of my food scraps and yard scraps. A few years ago, I really tried to boost my native bee population. I put up those bee tubes for the mason bees, and that worked really well. And then about 1.5 years ago, I moved. So there's my new setup, and really happy to have the deer fence because I struggled with the deer in my old garden. So I don't have to worry about the deer now, but I got a lot more rabbits and groundhogs living out in farm country. So I've got really poor soils, shale, clay-based soils. So I've had to go with the raised bed type gardening for both properties that I've been at. So, but what we're going to talk today is about soil biology. I went to school for biology, and I'm really excited that there's a lot of new emphasis around soil health because so much of these soil properties and our nutrient cycling and our disease and pest resistance really starts with that foundation of soil health and all these wide diversity of microbes that we have living in our garden soils. So this is a really complicated slide. I'm not going to go through it one by one, but you can see that there's a lot of life of all different types and sizes that we have beneath our feet as we're walking, and our hands are kind of working those garden soils. So we've got bacteria, we've got different types of fungus, nematodes and predators and earthworms and insects, right? That all kind of work in harmony and in synchrony in these good, healthy systems. And at the core of it is those plants on the system. And we'll dive in a little bit deeper. I put this up there just to give a little bit of perspective. When we're talking about soil microbes, bacteria are the largest functional group in our soils. So by weight, there's bacteria dominate our soil microbes. And then you can see that next level up, we've got the fungus species. We have archaea, which are kind of single cell, somewhat similar to the bacteria. And then like at the top of every food chain, we have the predators. And these are called protists, are the general family. So when we're talking about these soil microbes, they're right. We can visualize worms. We can visualize some of the bugs that we see crawling around. But these soil microbes and the fungus is tough to visualize, right? We don't all have microscopes in our backyard. So I've got some videos worked in with the talk. So I used to have this trick to be able to download YouTube videos. This one, don't tell the YouTube police, but I just held up my phone to the computer screen to record this video. But it is going to show us fungal filaments with bacteria moving in real time. This isn't sped up. This is real-time footage of how quickly that some of those bacteria are using these fungal networks through the soil as literally kind of like that road system, that highway system. So you can see as it's panning out, you can see the fluid that's flowing through that fungal filaments. And then as it pans out, you can see also some stationary bacteria that is not moving. It's fixed on those fungal filaments. And that fungus serves as that kind of highway network to move water, to move nutrients to our crop roots. But then it also hosts many different types of bacteria that also work to acquire those nutrients from our soils. So living plants are what drives this system. These microscopic creatures that live in our soils, they can't capture the sunlight energy. They can't make their own food. So they rely on that photosynthetic process of having plants in the garden, because plants are, the primary thing on earth that have that ability to transform that sunlight energy into simple sugars and then onto amino acids and proteins and all those other complex structures that filter through, life on earth and all the food chains that we drive about. So we're going to dive into a little bit about 4 principles for soil health, but of the 4, I really believe that having a living plant in your gardens as many months out of the year is one of the most important principles in driving this system. So with roots are really where the magic happens. So back to high school biology class, right? Plants can take sunlight energy, they take carbon dioxide from the air, water, and they convert that into simple sugars in glucose. And then as nitrogen added to that glucose becomes an amino acid, and you put a couple different amino acids together, and then you get that protein structure. But plants actually put a tremendous amount of those simple sugar products that they generate through photosynthesis down into the root system. So not only are they kind of building that structural stability in the root system itself, but they leak out. A big portion of these sugars get leaked out through the root system. We call them exudates. And it's these liquid sugars that leaking out of the roots that feed the bacteria, they feed the fungus, and they really drive this whole underground community. And most of the life in the soil is going to be concentrated right along that root zone. And it doesn't extend very far from the root itself. It's only a couple millimeters, so about the thickness of a penny. But you're going to have about 2,000 to 5,000 times more microbes living right along that plant root system as you would find in a lot of the other parts of our soils. And feeding that microbial system year round is really critical, because so much of this soil function, right, about 85 to 90% of soil function is biologically driven. So we want to feed those guys. So here's kind of just a graphic kind of illustrating those simple compounds leaking out. The growing root tip is one of the most active places of that root system. And at different growth stages, plants are going to leak out different amounts of those growing sugars. So when the plant is actively growing, right, it's putting on a lot of top growth. That is also the time when it's putting out a lot of these simple sugars, building the root system and acquiring all of the water and the nutrients it needs to do that top growth in the framing stage. But once it starts kind of producing fruit, setting seed, it gets into that reproductive stage, it's pretty much acquired all the nutrients that it needs. Most of the water uptake is still happening, but at that point, when it reached maturity, that system kind of shuts down, and there's not going to be as much of these sugars leaking out of that root system in the late summer, kind of early fall, when those plants have reached their full growth potential. So this is what those roots exudates kind of look like. Here's a microscopic image, these droplets of water. And what's unique is that each individual plant species has slightly different chemical compounds in these root exudates, and that attracts different types and different species of these soil microbes that serve different functional roles. So the image on the left with the inflorescent green, right? The middle part is the actual roots. And then they dyed the bacteria this fluorescent green, and you can see the concentration of bacteria right around that plant root zone. So we want diversity in the system. So the more types of plants that we have growing in the garden systems, the different types of root exudates they're going to be putting out, and then we're going to have a higher diversity of those soil microbes living in the soils. So why are plants going to go to all this effort, right, and waste a lot of their photosynthetic products out into the root zones? And it's because of these bacteria, and these are the primary feeders on the sugars. So the bacteria live right along the root zone. So the sugars are feeding the bacteria. And then we need the predators in the system. So these are ciliates. They're one of those predators. And again, they dyed that bacteria green. So you can see on the inside of this single cell organism, all of those different bacteria that they've eaten. So the protists are eating the bacteria. And then in that process, they're consuming extra nitrogen. And all that extra nitrogen gets secreted back out into the soil next to the plant root. This is what mycorrhizae fungi looks like. And this is a good beneficial type of fungus that grows right into the crop roots zones. And it really extends that root network. So we've got mycorrhizae fungi that's eating these sugars. That mycorrhizae network is going out and collecting nutrients and water, and then bringing it back to our growing crops. So it's really beautiful cyclical system that we've got happening here. So again, here's another video that shows, this one has some narration.
If you'll notice, it grows through the soil.
Can you guys, Kim, can you hear that narration with the video?
The sugar.
Yes, it's a little low, but you can hear it.
Fertilizer is not plant food. Plant food is sugar. As cells slough off, as you see, they are consumed and inhabited by bacteria. The bacteria are part of the plant. They're in the plant, on the plant. They're supposed to be there. While you can't see them, the liquid around this plant root and these hairs is filled with nutrients.
All right, so I'm going to stop it
And
here,
that's
just
all
in the
about
essence of time. But those are those little predators that are swimming around, and I've got another video coming up. But one of the things I hope that illustrated is there's a lot of movement and activity in our soils, and we don't often think of our soils as an aquatic environment, but really it's important to have that good soil moisture, to have that thin film of water along these soil particles, along that root zones, because a lot of these microbes in our soils rely on that water type environment to be able to move through the system. So bacteria, I want you to, oftentimes, right, bacteria has this negative connotation, but there are so many beneficial forms and beneficial outcomes that we get from bacteria in our system. So I want you to kind of shift your perspective a little bit with bacteria, and in the agricultural setting anyway, think of them as like little bags of fertilizer that we have in our soils, because bacteria are the thing that functions best at acquiring available nitrogen. So if you're fertilizing your crops during the summer, you may think that that fertilizer is getting directly into those crop roots and into your plants. But in reality, a lot of that nitrogen fertilizer is first being consumed by the bacteria. Plant roots actually are very inefficient at absorbing nitrogen directly out of the soil profile. So they're tying it up, and then it becomes a nice, stable form of nitrogen. The problem we have with nitrogen, especially the nitrate form, is it dissolves real easily in water. So if we have a lot of extra nitrogen in our systems, we get a good heavy rain, or you do a real heavy watering maybe once a week, that's really got the potential to flush that nitrogen down below the root zone, and not be crop available anymore. And the other amazing thing about bacteria is they can respond and grow their populations really quickly. So under the right conditions, they can double their populations in 15 to 30 minutes. So they can respond really quickly to those changes in the system. And these are what some of the predators look like. So we don't want all that nitrogen tied up in these bacteria, right? Because then our plants are going to have yellow leaves and they're going to show some nitrogen deficiency. So this concept of mineralization is really comes down to how much of this predator population we have. So these things I think are just really cool. These are some scanning electron microscopes of some of these protists in the system. So some of them anchor themselves and stay in one place, and then they have like these filters to bring in the bacteria. And then some, like we saw in that previous video, move around and actually go after. We also have nematodes in the system, and nematodes again are one of those critters that have more of a negative connotation, right? More of those pest nematodes, but there's a lot of kind of just neutral nematodes in the system, and these predatory nematodes that are also eating the fungus, eating the bacteria, and cycling those nutrients into our growing plants. All right, so here's what those protists look like as they're swimming around. So here's one of those. Now he kind of looks right now like he's anchored, but he's not. So he's going to start moving around, but you can just see again, when they've got the proper conditions, how active they are with swimming around. So if you want to dive a little bit deeper in your studies, learning that relationship with carbon to nitrogen ratio is time well spent. And I encourage you to understand a little bit more of the nuances with that. But if you've got, say, some compost that you're adding, or some fresh leaf material or mulch that you're adding into the garden system, how quickly that mulch material breaks down is going to depend on its carbon to nitrogen ratio. So like straw mulch has a lot of carbon and a little bit of nitrogen. So that breaks down a lot more slowly than, you know, if you're weeding and you're just putting kind of fresh green material down as a temporary mulch, that fresh material with a lower carbon and nitrogen is going to break down a lot quicker. So this carbon to nitrogen ratio comes into play with how this nutrients and nitrogen especially is cycled through this mineralization process. So bacteria have 5 carbon for every 1 nitrogen in their bodies, but these predators have a higher carbon to nitrogen, right? So they need 30 carbon for every 1 unit of nitrogen. So in order to meet their nitrogen needs, those predators need to eat 6 of the bacteria to get their 30 carbon. But in the process, they are consuming all this extra nitrogen that they don't need. So they excrete that. It's like protozoa poop. It's in the ammonium form of nitrogen, which is really easy for plants to take up that ammonium form. And then that becomes that mineralization process to feed our plants and crops. So fungal feeding nematodes also have even a higher carbon to nitrogen ratio, right? So we're getting even more of that nitrogen cycling through that biology in the system. So another way that we can add nitrogen into our systems is by having legumes. So legumes, right? That's going to be our peas, our beans, a lot of our clovers. And it's pretty well known that legumes add nitrogen into the system, but it's not really the plant itself. It's these family of bacteria, rhizobium bacteria, that form associations onto these legume crop roots and form these little nodules, and they house nitrogen fixing bacteria. And they've got the ability to take nitrogen gas from the air and convert that into nitrate, which is a form that the plant roots can actually absorb. So if you're planting peas or beans, if your seed catalog that you're ordering from has the opportunity to also order an inoculant, I would definitely encourage you to spend a little bit of extra money and inoculate your peas and beans and any of the other legumes you're planting in the garden, because that's that dry, dormant form of the bacteria that's going to colonize these roots, right? And that's really going to pump a lot of extra nitrogen into your system if you've got that inoculation as you're planting those seeds. So we can get nitrogen for our garden from free with these nitrogen fixing bacteria on legumes. But then we're also learning that there's other types of nitrogen fixing bacteria that live freely in the soil. They don't need to form that nodule on the crop roots to be able to fix the nitrogen. So as we get more diversity in the system and as we kind of build this good house for the soil microbes, we're probably going to have some of these free living nitrogen fixing bacteria that is going to feed our crops as well. And then this is really cool. So this is kind of a new process that's really only been identified in about the last 5 years. And James White out of Rutgers has really been leading the research on this process, but it's called rhizophagy. And this could potentially be a large way that our plants acquire their nutrients from the soil, right? If you don't, especially when you don't have that fertilizer in the system. So what happens is at the growing tip of that plant root, right? It's leaching out all those root exudates and those simple sugars. And then just beyond the growing root tip, where this image on the left says absorption zone, the plant root actually eats the bacteria and eats the fungus out of the surrounding soil and absorbs it directly into the plant roots. So it eats the bacteria, it strips the outer shell off of that bacteria, and in the process, it's getting a lot of these micronutrients, right? It's getting manganese and iron and calcium and magnesium, but then it's also got the ability to strip some nitrogen in the form of nitrate off of that bacteria. So some of the bacteria die in this process, but a surprising amount actually survive getting their cell wall stripped apart. They turn into these little round cytoplasts, and they actually secrete a plant growth hormone that then triggers the root hair development. So all those finer root hairs that we get coming off of the main roots get stimulated to grow more root hairs, and these partially eaten bacteria travel down the root hair. And when they get to the growing end of the root hair, they get spit back out into the soil to live another day, and they form a new shell around them. So you can tell I'm pretty excited about that. I think it's really cool as we're learning more of these natural processes of how these plant roots interact with the soils all around them.
Hey, Lisa, can we
Yep.
pause for just a few questions? I
can.
see some stuff in the chat box, and I didn't know if anybody needed to come off mute and just ask a few questions before we move on.
Yeah, I'm happy to take questions. It gives me a little break from talking. Or
So
if you wanted to read.
Michelle had made a comment that she's looking for beneficial nematodes. So I didn't know if you wanted to comment on that. And then there's one more. I don't know if there's a source that.
That is one of the things I've not yet tried in my own garden, is to purchase any of those beneficial nematodes. So if you've got kind of a trusted source and you've done your research and you think it's a pretty reputable company, I certainly don't think it would hurt. I think the nematodes that are being sold by Kodak have been pretty well researched and fleshed out that they truly do have that beneficial nature. But I'm sorry, I don't have any personal experience with adding nematodes to the system.
I had a question on the nematodes themselves. From companion planting, the marigolds, they, well, from what my understanding is, they can produce chemicals that can produce the types of nematodes that can help with the root knot nematodes, or some type of substance that helps with root knot nematodes.
I would not at all be surprised at that because, like I said, those different plant species can produce kind of those different chemical signals that can either enhance or suppress some of those microbes in the soil. So
Okay.
I think that's very possible that interaction is happening. Yeah. Yep.
And then the second question I had was actually like a multiple question. When you talked about the overwatering, leaching out too much nitrogen, is that really happens when you overwater? You wash all your nitrogen out of your soil, so it's best to let it dry
Potentially.
out some before you keep watering.
Well, I think, right, the trick is not to have too much extra nitrogen in the system to begin with.
Okay.
And, you know, that's going to be a learning about watching the color of your plants, because sometimes actually too much nitrogen, you'll get too much vegetative growth, and then your fruit development you'll get too much leafy growth and not as much energy getting put into fruit development in a really high nitrogen system in gardens. So yeah, but even heavy rains, when that soil gets saturated, it's got the potential to lose some nitrogen out of the system. Yep. So finding that good balance of just enough, but not too much, that's going to be lost.
And then my last question was, can you explain more about what inoculation is and why it's important when you're planting certain seeds?
Yeah. So like I said, the legume species, so the peas, the clovers, beans, anything in those families have the ability to form this relationship with that rhizobia bacteria. So when you're purchasing an inoculant, it'll be typically like a black powder that's in a sealed bag, and that actually contains that rhizobia bacteria species. So you can cover your seed, right? Sometimes you want to do it a little sticky. If you're having trouble getting it stuck to your seeds, like peas and the bigger stuff, you can actually roll your seeds in like a little bit of syrup or soda, right, to get it a little sticky, and then sprinkle the powder and roll them in the powder before you plant them. So that way, as soon as that seed sprouts and that new little root starts growing, that bacteria, that nitrogen fixing bacteria, is immediately right there on the root zone on the seed to start that colonization and that nitrogen fixing process. Yeah.
Okay, thank you.
Yep.
I have a question about the rhizobium bacteria as well. How long does that last in the soil once that crop has been rotated out? Like if you are pulling peas out and you plant, you know, cucumbers, and then in the fall you bring back some beans, like what is the timeline for the existence of the bacteria without a legume to pair with?
So my understanding, bacteria, when they've lost their food source or the conditions change, they can go dormant pretty quickly, and they can remain in that dormant stage for quite a while. So if you've got that period of a gap where you don't have a legume or a host species, they'll go into that dormant stage, and then when you do have that appropriate host species secreting those root exudates, that's going to stimulate those bacteria to break out of dormancy and potentially colonize again. I think the harder part is when you plant that new seed, it's going to take a while potentially for that new root seedling to grow in that particular area of the soil where that dormant bacteria may still be living. So by spending that little extra time and extra money on the inoculant, so you're guaranteed to have that rhizobia right there on the seed at germination, you'll get a quicker response and probably a higher colonization rate with the inoculant than you would dormant bacteria in the system.
Lisa, there is one more question about your opinion on azomite or rock dust.
I'm not familiar with the first one. The rock, so I'm assuming they're both used as kind of a mineral supplement, right? That's for the micronutrient supplements of the rock dust? And something might help clarify that for me.
Avery, did you want to come off mute and clarify your question? I'm assuming as well, but
Yeah,
maybe Avery
so
can clarify.
Yes, yes. Hello, can you hear me?
yes.
Okay, so yeah, so with my garden, I do add azomite, which is a particular brand of rock dust. I just wondered if you had an opinion on that, because some people say it works, people say it doesn't. Yeah, there are certain things in the soil to help facilitate those minerals being uptaken by the plants. So yeah, if you have an opinion on or science or heard anything about how well it's used, or if it's used at all. I mean, if it's used well at all, if it's successful.
All right. So I have not heard any negative reactions or consequences of the rock dust. One year in my garden, I bought green sand. I think for that's kind of same purpose. It was that good kind of natural mineral-based material. I think the challenge is, right, it's still in that kind of rock form, so it's not going to be immediately available that first season or maybe even the second season. It's going to have to slowly be decomposed and broken down for those rock minerals to become plant available. So I haven't heard any negatives, but if you're applying it and expecting some results that immediate growing season, you may not get that immediate response. But I haven't, there's not many issues. When micronutrient levels get really high and really out of whack, sometimes they can compete with other nutrient absorption in the plant. But if you're doing it in moderation, I don't foresee any problems with it.
Thank you.
Yep. All right, Kim, I'll rely on you. Keep eye on the chat, and I'm also keeping an eye on the clock, right? Because it's fun for me to talk about this kind of stuff. This is an example of what the mycorrhizae fungus looks like. So again, mycorrhizae fungi is a beneficial fungus. It is synergistic, so it grows actually inside the root system itself, and then really expands the root system of that plant. So here's kind of a microscopic image. These are kind of the spores, the reproductive stage of that mycorrhizae. And then sometimes when you are digging, every once in a while, you'll be able to see kind of a collection of these filaments into a hyphae, and you can actually see them sometimes when you're working the soil. So plants at a very, very young age, almost immediately on germination, form the relationships with these soil microbes, especially when we don't have too much fertilizer, too much inorganic kind of forms of nutrients in the system, and they rely on these microbes to acquire their roots, right? So they form the relationship with the mycorrhizae fungi because the root hairs are large. They don't actively produce enzymes to go out and acquire their nutrients. But these mycorrhizae fungi do produce these enzymes to go out and get nutrients, and they host those like phosphorus solubilizing bacteria and other forms of bacteria that also have the ability to go out and acquire nutrients directly from the soil. So the fungus is a lot smaller. So when you have that relationship with your crop roots, it can kind of get into all those smaller nooks and crannies of the soil particles that are too big for the roots or even the root hairs to access. And this relationship really evolved to supply phosphorus to our plants, right? So kind of the big three for fertilizers that we need are nitrogen, phosphorus, and potassium. And plants that associate with this mycorrhizae fungi are really, really efficient at acquiring the phosphorus that's out in the system. They're great with water uptake, and it binds with calcium. So sometimes when you get a really high mycorrhizal soil, your pH tends to steady out, and you don't need to be adjusting your pH with adding lime as well. But especially in a gardening situation, not all of the plant families form this relationship with the mycorrhizae. So most plants do, but some of our garden plants unfortunately do not, and we don't know why that is. But anything in the brassica family, so down at that last bullet on the slide, so any of our canolas, our kales, our radishes, anything in those brassica families don't form this relationship, as well as, you know, some of the beets and the rhubarbs and stuff. But in general, most of our plant families do, including weeds, right? So if you've got some rows and you're kind of strategic about where you're planting these brassicas or these other crops that maybe don't associate, perhaps being strategic and kind of alternate your planting so that in between those brassica rows, you do have a plant that is going to be a host plant for that mycorrhizae network. So you can still expand it. So oats are really good. We're going to talk a little bit about cover crops later in my presentation. But oats are an excellent cover crop and highly mycorrhizae. Sorghum and sorghum sudan in the system as well. Typically in a gardening type situation, we're not using much of that sorghum, sudan, and crops, but oats is going to be a really easy one for you to incorporate as a cover crop in the garden. So here's kind of what that network looks like. Like I said, it's really good at finding phosphorus. And I'm going to kind of go through this slide just in the essence of time. We are finding in high phosphorus soil, so like in a big row crop setting, chicken manure has a lot of phosphorus in it. So some of these farms that have been spreading chicken litter on their fields for 20 or 30 years have really high phosphorus. And when phosphorus levels are high in your soils, the plants are not going to bother forming this relationship with the fungus because they can acquire all the phosphorus that they need. So we don't yet know where that high phosphorus level threshold is for when this relationship with the fungus breaks down. So it's not only phosphorus that gives us a challenge when we're talking about these soil microbes, but also extra nitrogen in the system can inhibit the plant roots putting out the energy and forming these relationships. So this is a slide from Christine Jones. She's out of Australia, really brilliant lady, but she travels all over the world. And these are some of her observations that she's seen with oat roots and oats that were being grown in a real high nitrogen fertilizer situation. You can see on the left, they didn't form the association with the soil microbes versus the image on the right in a lower nitrogen situation. The other thing that we benefit from supporting all this biology in the system is that's what gives us our good, stable soil structure. So when you're digging in the garden, right, you want that good dark brown, high organic. You want that soil to be crumbly like cottage cheese. And it's really these biological glues that are secreted by the bacteria, secreted by the fungus, that really creates, we call them aggregates. And it really creates that good crumbly soil structure with lots of like open airspace and pore space, right? For the water to soak in and the air to get it down into the root zones. So these red arrows on the one black and white picture kind of highlight those biological glues that hold those soil particles together. And then the image in the middle is that mycorrhizae fungi network, right? That spider web network in the soil that can physically wrap itself around these soil particles and create that aggregation in soil structure as well, right? So this is kind of what it looks like. I think a great simple trick for you to do to kind of check how biologically active your soils are is to, you can do it with weeds as you're weeding. You can do it with maybe some crops as you're thinning them. But as you're pulling up the roots, the plant by the roots, shake off the extra soil. And if you see this image like on the top right with kind of like those dreadlocks, right? Those soil particles that are attached right along the root and they don't shake off, then you know that you've got the biology working in your system and your plant roots are forming those relationships with the soil microbes. So that little image up there, that was a 8-day-year-old rye cover crop seedling that I dug up. And you can even tell after only 8 days how much relationships and how they've binded with the soil. All right, the other great thing about having microbes in your system is that's going to lead to that really good long-term stable organic matter level, the humus component of organic matter level. So we often think that by having a mulch layer on the top or adding kind of some fresher compost, that's a lot of the ways on how we can build organic matter levels in our soils. And it's a lot easier for us at a smaller scale in a gardening type situation to really put that additional organic material in the system. In a row crop, 500-acre field, it's a lot more difficult to have those heavy inputs of organic compounds. So even that mulch layer that's left on top and is slowly breaking down, most of the carbon that's in that residue that's left on top actually, as it's decomposing, gets respired up as carbon dioxide back up into the atmosphere. So really only about 15 to 20% of the carbon in that kind of old plant residue mulch that's left on the surface makes it into that long-term kind of stable humus part of it. So it's really the dead bacteria, the dead fungus, and those cell body structures that form a large component of the stable organic matter level, right? That we want, right? Because it acts as the sponge and it holds on to those nutrients in our soils. The other thing you can keep an eye out for, if you see some mushrooms growing in the soil, that's another good indicator that you've got a lot of good of this saprophytic fungus, right? So this is different than the fungus that grows into the crop roots. This is the type of fungus that decomposes things, right? So if you peel the bark off of a rotting piece of firewood or something and you see that fungus. But we want that other type of fungus near the surface of our system to help break down the residue and break down some of those fresher organic materials as well. And then earthworms, right? Everybody loves earthworms. I love finding them as I'm digging in my garden, but earthworms are Mother Nature's tillage, right? And when you've got a good population of earthworms, man, they can really work the top 6 to 8 inches of that soil and keep it aerated and be cycling those nutrients. The earthworms are going to be responsible for moving bacteria to different parts of your soil, some of those spores for the fungus, and they're really kind of distributing those microbe populations throughout your soil as well. All right, so oftentimes we've got multiple types of worms that live in our soil systems. These are nightcrawler type worms. So in a gardening type system, I know I have not really seen these types of worms in my home gardens. Perhaps some of you have, but these worms live their entire life in one burrow. They only come out at night and they stretch out of their holes. And like you can see that they're doing here, they're going around and collecting those surface residues, dragging them back into their holes, and then they're eating and processing that above ground residue. So sometimes later in the growing season, later in the summer, you can walk a field and see these little clusters of old straw residue or old residue, and usually underneath each one of those are one of these nightcrawler holes. But like I said, I don't often see them in my garden situation. I get more of what I call these kind of classic fishing worms. These worms are a different type of worm. They're going to move in all different directions through that soil profile, up and down and sideways, and really like create those good channels for the roots to get a little deeper, to aerate our soils. The other third type of worm are kind of the red wigglers. So if you've ever done any vermicomposting, these would be kind of those red wiggler worms that you'd use in the worm composting. And typically we'll see them more in like a forest situation, leaf litter type situation. So we want this system to be working, right? We want this good worm population to be working the soil. We want good soil structure. We don't want to have these soils that are really fine and kind of pulverized to create this crusting. So oftentimes if you're not kind of cultivating between your rows, and we get a good heavy rain, oftentimes you'll see kind of the soil crusting, because the power of a raindrop hitting bare soil has a tremendous amount of energy. And a lot of our soil moves and is eroded by that initial impact of that raindrop hitting bare soil. And the other thing that happens is we get this crusting. So what that really inhibits is our soils need to breathe. Our crop roots need the oxygen. These soil microbes need oxygen deeper in the soil profile. And as the microbes are eating that residue and cycling these nutrients, they're respiring carbon dioxide just like we do. So we need our soils to breathe. And anytime we get the soil crusting on the surface, it's preventing those soils from breathing. The other thing that's really critical is we want to keep our soils in kind of that happy zone, right? Earlier I talked about the importance of soil moisture, because a lot of these soil microbes are aquatic organisms, but they also have this real sweet spot for soil temperatures. So our microbes are like this time of the year, they're just starting to wake up. They've been dormant all winter with cold temperatures. They kind of shut down. But when our soil temperatures really start getting around 50, 60 degrees, those microbes get really active again, and they're looking for a food source. And again, I'll show you some examples of cover crops, but that's one reason why we want to have cover crops planted in the fall and have some types of cover crops that survive the winter and are there in the spring. So we have that food source, those sugars, those root exudates for these microbes as they're breaking dormancy in the spring as the soil temperature warming up. So that activity is going to double for every 10 degrees that soil warms up, but the sweet spot is really between about 86 to 95 degree soil temperatures. You definitely want to avoid anything above 95 degrees because then we start losing a lot of moisture through direct evaporation out of our soils. And it really becomes kind of a moisture control issue. So when we're talking about soil health principles, you'll see sometimes a graphic where you have 5 principles and sometimes even 6. For illustration purposes, I like this graphic of 4 main principles. So we're going to be minimizing disturbance. We used to focus a lot on tillage and rototilling and actual soil disturbance, but we now understand disturbance also includes those chemical inputs, and those fertilizer inputs is also a form of disturbance to our soils and our soil microbes. We want to maximize the soil cover. Like I said, we don't want that bare ground for that raindrop or your hose water, right? To reach that soil surface directly. You always want to maintain that soil mulch, to keep those soils cool, to retain that moisture and break up that energy of the raindrop. We want to maximize diversity in the system. So that could be through crops, through crop rotations, through cover crop mixtures, adding some composted manure sources, lots of ways to build diversity in the system. And then here's that key principle that I think is one of my favorites is maximizing that continuous living root system as many months out of the year as you can. Right. So those principles are really going to influence the structure of your soil and how well your soil structures. Because you can be farming on soil or you can be farming on dirt, right? Depending on some of those management decisions that are being made, right? So tillage is going to do a couple things. If you still feel like you haven't built up your soils enough, so you got that good, fluffy, aggregated soil, and you still need to do a little tillage, I just want you to understand you're going to set yourself back with each time you do a tillage event, right? We talked about that fungal network, that spiderweb network that builds itself through the soil. When you're going through and tilling, heavy spading, heavy digging, you're literally severing and cutting like the arms and the legs off of that fungal system. And it's got to put all that energy into reestablishing itself during that growing season. The other thing that happens as we're tilling and we're introducing oxygen deeper into the soil and into parts of the soil that were maybe low oxygen levels, it stimulates this copatrophic family of bacteria, and that population explodes. Remember, I said bacteria can respond really quickly, 15 to 30 minutes under the right conditions. So by introducing oxygen, you're stimulating more of this certain family of bacteria to grow. They're looking for that carbon source, and they're going to start eating that organic material that you've been trying to build up in your soils, right? And I put this graph in here that shows how much carbon dioxide, how much of that carbon that's respired as these bacteria are eating that carbon in the first 5 hours after a tillage event. And you can see a full moldboard plow event. You got 81.3 units of nitrogen or carbon dioxide loss compared to a very reduced till, no till type disturbance, right? So there's a big loss of carbon in that first flush. All right. So in the last 5 minutes, and Kim, I'm sorry, we're running a little late. I'm just going to show you
Sorry, if you need a few more, it's fine.
I'm going to show you some examples of how I am managing my garden. So I no longer like using any sprays in my garden to kill my cover crops. My beds are small enough that I can go in and mechanically cut my cover crops. So I just use this big like mechanical hedge clipper type tool. So this is me and my system for planting peas like early April. So I'm clipping my cover crop in the spring. I should have brought it down. I have this old-fashioned kind of narrow gardening tool that's about 3 inches wide, probably, and it's like this small cultivating kind of tool. I just scratch a surface, a trench right through my cover crop roots. It sometimes takes me a while to kind of dig through the root masses and pull out the roots. But I'm digging that real narrow, probably 3-inch wide trench in my raised beds. And then so again, there's some example of what it looks like when I pulled up my cover crop roots. So then I'm just laying my peas right down that freshly kind of scratched out trench. I found a chopstick works really well. I just kind of like do a little soil disturbance underneath my pea drops right down into my hole. And then I got that nice loose soil to cover it up. What I have learned for planting peas, when you try to mechanically cut a cover crop when it's still in that young growth stage, it's not going to die by just chopping it off like this. It's still in the growth stage. It's going to regrow. So what I do is I lay a couple sheets of newspaper down, and then I have a dairy friend who has some kind of composted dairy manure that I can get, but any sort of like carbon input or manure or compost that you may have. So I lay that compost directly on those sheets of newspaper, and that does a really good job of kind of smothering out and killing the rest of my cover crop in between my pea beds. And there's my peas, I don't know, probably early June sometime. But you can see I got pretty good weed control with that newspaper and compost trick in between. The other thing I do, and my system is really slowing down now. I don't know what's going on. Later in the season, right, as my cover crops get a little taller, they're going to kill a little bit easier. So here you can see I had some crimson clover that survived the winter that year. You can see the red blossoms. I'm still going and mechanically clipping those. And then if I've got zucchini or squash or anything that's already in a transplant type state, I call this planting green, right? So I'm just digging a hole about twice as big as whatever my transplant pot is, and only disturbing that small amount in my raised bed to plant my transplants. And then I'm using this old cover crop as that mulch control in between. This one, you can see I didn't, you have to go back and do some secondary trimming of some of these cover crops. I have a feeling this is the summer when I made the huge mistake of having annual ryegrass in my cover crop mix. Please, please, please learn from my mistakes. If you're going to be kind of experimenting with some cover crops in your garden, make sure you do not include anything that has annual ryegrass. That's different than cereal rye. So this is annual ryegrass. It becomes a weed. It continues to grow all summer, and it gave me trouble. So I have a suspicion this was the summer. So it's a little messier style of gardening if that's something that you can live with. That's kind of up to you. So preparation is going to start in the fall, right? You want to get a good stand and a good catch on these cover crops in the fall. So as I'm cleaning up my beds in the summertime, I'm also at that point. So first, let me back up. I don't pull up from the roots. As I'm cleaning up my beds, I'm clipping the stem right at the soil surface, and I'm leaving that root system intact, right? I don't want to disturb that soil. I want that other kind of root mass, organic matter in the soil to break down and add to my organic soil organic matter. So I'm clipping everything right at the soil surface. My raised beds always seem to sink. I have no idea where the soil goes, but every year I feel like I still need to add some soil or some compost. So I'll put a kind of a fresh thin layer on the top, build up the bed a little bit at the end of the growing season. And then I'm just broadcasting my cover crop. I always like to do a mix. I'd like to have oats and buckwheat because they grow really quickly. But then I also like to have some things that will survive the winter, because up here in Pennsylvania, if we get a couple hard frosts, the oats and the radishes and the buckwheat will die out pretty quickly. So anyway, I just broadcast it. You can see my metal like stone rake that I have there. And I just kind of lightly scratch like maybe the top inch to work that seed and get some seed to soil contact. I try to do this and time it before I know a rain is coming. So then I get better seed to soil, and I get that kind of flush of water with germination. But you can water right after seeding as well. So again, the more seeds in the mixture of my cover crops and the timing of it, this picture on the bottom, you can see that year I cleaned up the beds in very different stages. So this is the same exact cover crop mix, but you can see how much additional growth I got and how differently it expressed itself when I had the first bed planted at the end of August versus like probably 3 weeks later, mid-September, and then early October. So I encourage you to, as you're cleaning out the beds, try to get these cover crops planted as quickly as possible. The other thing I like with the cover crops in my garden is I really think it helps with my weed control, especially like right now that the flush of those spring weeds. So the purple dead nettle, kind of the chickweed, the henbit kind of stuff. But typically when I have a cover crop planted, I get much better weed control. You can see in this corner of my raised bed, I had a rosemary plant that was still growing in the fall and it lasted half the winter. So I didn't get cover crops established underneath that rosemary plant. And you can see that's about the only spot where I had spring weed issues. And then if you're planting later in the season, right, you can let that cover crop grow. You can see that's almost as tall as me right there, that rye, cereal rye that I let grow. I'm still cutting it. Actually, my husband helped me out last year and he got the mechanical hedge trimmer and helped me cut this really tall stuff. But I'm basically growing my own mulch by doing this. So I'm cutting it off when it's real mature, that high carbon to nitrogen ratio. I just leave it lay alongside my bed and dry out. And then I've got that to also kind of feed in and add some extra mulch layer in between my plants. All right, I think I'm almost done here. Let's see. Oh, the other thing I like having with the mulch, right, is it creates that benefit, that good habitat for the beneficial insects. Right. So I love seeing the spiders in my garden. I love seeing these ground beetles. Sometimes with this type of garden, you have slug issues. And these shiny black ground beetles are really good slug predators. And then lightning bug larvae are also really good slug predators if you're struggling with that in the garden. But creating this habitat, right? I know it looks messy and it's not a neat and tidy garden, but having that kind of messy habitat is great for the beneficial insects. So again, think of ways that you can add diversity into the system, whether that's homegrown compost, whether you can get some manure source, some mushroom compost, and then also just diversity, not only in your vegetable crops that you're growing, but do you have the opportunity to do some pollinator plantings around the gardens or near the gardens, right? To bring in those beneficial insects and have those different host plants for those beneficial insects around the garden as well. All right, so just to recap, these are those 4 soil health principles that, have those in the back of your mind, right, as you're managing your garden for this next growing season. So you're going to try to minimize that disturbance, keep those soils covered, keep that mulch layer, keep them protected, try to get as much diversity in the system as you can. And again, have as living roots as many months out of the year as you can, right? And this can be a whole nother hour-long talk, but I really believe that the healthier we make our soils, the more diversity, the more micronutrients we get, that's going to translate into nutrient-dense foods, right? And we've got this huge opportunity right now to be growing better quality food and better quality crops, changing our diet habits to really address and start using food as medicine to address some of our health concerns. And that's really my next area of interest, right? Is kind of this whole nutrient density, food as medicine type conversation. All right, so this is my wrap up slide. So we spend a lot of our time and energy in the summer actively managing our above ground part of our garden, right? Managing the plants, pulling weeds. But hopefully by this talk, you recognize you've got responsibility also for these microbes and all of this life that lives in the soil that is also dependent on these management choices that you're making. And it's really these soil microbes that are driving this good, healthy, functioning gardening system that we're all trying to manage. So, okay, that's my last slide. So I will stop sharing, and I'm sorry we went a little long, but happy to take some questions.
It is okay. That was awesome, Lisa. Thank you so much. And I think we're going to have a couple of questions. So I'm going to turn it over to first, it looks like Melanie.
Hi, thank you for sharing tonight. This was definitely really good. I saw the line for you after your session at Future Harvest. So I was like, what's all that about? So thank you for sharing. I too made the mistake with winter rye, and it was an absolute traumatizing nightmare. So you mentioned cereal rye. So, well, the good thing that came out of it was I was able to use it as a mulch once it dried. But I don't ever want to use it again unless I'm on a much larger scale and can terminate it, in an easy way. So can you tell the difference between using the cereal rye? It seemed like maybe it was better.
Versus the annual ryegrass.
Yeah.
Yes. Cereal rye is one of my second favorite, right? I mentioned how much I like oats. But if you've missed that fall cover crop window and it's later in the season, cereal rye is really about your best option for planting a cover crop later in the fall. Because it can germinate and still grow at like soil temperatures down into the 30s. It's a really amazing crop. So I would still encourage planting cereal rye, even if it's later in the season. You may not get a whole lot of top growth and the benefits, over those winter months, but that seed's going to be germinated and ready to grow in the spring. As those soil temperatures are warming up, like, this time of the year, a lot of the fields that I'm driving around is cereal rye. So again, it's going to be about having that early spring food source to kind of drive this biological system. So rye triticale is another kind of later season option. That's a hybrid. It's between wheat and rye, is called triticale. That also can be planted later in the season. It'll survive the winter, and it kills off pretty easily. So the cereal rye, the triticales, any of those small grains, the longer you can let them grow and mature before you terminate them, the better kill you're going to have and less regrowth. So.
Thank you. And then just one other quick question. Do you use a different mix? Do you use a cover crop in the spring? And if so, do you use a different mix?
So again, I come back to my oats because there's a couple types of spring oats that you can do. I haven't tried it yet in my garden because I tend to have clovers in my fall mix, but frost seeding a red clover could be an option for you if you're trying to get in a spring cover crop. That freeze thaw cycle, when you're broadcasting red clover at that point, gets you that good seed to soil contact. And I think you can get a pretty good catch in the spring with frost seeding red clovers as well. Yeah. And buckwheat too. I mean, if you've got a little bit of time, probably anytime after your last chance of a killing frost, because buckwheat is really susceptible to frost. But if you've got a late spring window, buckwheat is another real fast growing plant that you can maybe try to sneak in the springtime.
Oh, so
Great
so with
questions, Mel.
buckwheat, you just don't want to let it go to seed, right?
Right. It can it's a hard seed. So if it does go to seed, it could be there for several years, right? And vetch would be the same way. Some of the cover crop mixes have vetch. That's also a hard seed that doesn't always germinate, and you could have some carryover as well. But yeah, I don't know. It's diversity, right? Like, and I'm not
Yeah.
real aggressive with pulling weeds anymore because I kind of view weeds as Mother Nature trying to add some diversity into the system. But you definitely don't want to let them go to seed and set seed with the weeds. Yeah.
Yeah. 1 year I had an invasion of vetch that just came in from I don't know where. It took over. And it took me, I guess, a couple of seasons to get rid of all of it. But I didn't have any problems the following year. But when it came in out of nowhere, it just kind of just invaded the whole hillside. And it was crazy.
Well,
So
good for you for finally getting rid of it, because that's a
yeah.
challenge. Yeah.
Yeah, that's just a beast.
Any other questions for Lisa? Lisa, I had one. And I know there's a little bit of controversy about potentially with sheet mulching, to terminate cover crop in terms of inhibiting, gas exchange. I didn't know if you had any experience with that and if it is really an issue or if that's also a potential way where folks can terminate their cover crop, like through tarping or whether it's clear plastic or black plastic.
So I don't use any black plastic or plastic mulch in my systems. Mine are small enough. And I get a little concerned with the black mulch for a couple reasons. I do think I guess depending on the type of the plastic, right? You don't get that air exchange, right? So you could limit yourself to that. The other thing that concerns me with the black mulch is on those good hot sunny days, you could really be heating up that soil surface and potentially starting to kill off some of the soil microbes with those high soil temperatures as well. I know there's many pros of the plastic, right? It's a lot easier to manage and weed control, and you get better moisture retention if you got some drip irrigation underneath it. But yeah, I think anytime you can kind of keep a more natural material for your mulching, I would prefer to see that. But depending on your scale, I know that's not practical in a lot of cases.
You think even for I'm just thinking like maybe not for the whole season, but some folks will tarp for maybe a couple of weeks or a month or two just to knock down cover crop. I didn't know if that has too much of an effect on the microbiology or if just for like a month or so, if that's
so I think
doable.
I think that would be an acceptable method to terminate, right? I think it's a much better alternative than doing an herbicide termination. It's a lot less labor intensive, right? If you got a large area and you're trying to kill that cover crop, so it's a lot more practical than the way I do it. So no, I think temporarily, I think these systems are flexible enough to recover. I don't see that 3 or 4 weeks kind of really setting back the system too much. Yeah.
Okay. Thank you.
I have one last question, I promise. What's your take on cardboard for termination?
I think cardboard would work really well too. Maybe even sometimes a little better than my couple sheets of newspaper, right?
Right.
And you're adding that extra carbon into the system. So yeah, I've never tried the cardboard, but I don't see too many downsides to that either.
All right, thank you.
Any other questions for Lisa besides when she's coming back for a part 2? I see a few people mentioning that.
I do.
All right. Well, thank you so much, Lisa. That was really awesome.
Yep, I enjoyed it. Happy to be here.
We enjoy you sharing your passion with us. It comes through. And I don't know if anyone else on the call has any announcements. I think I had mentioned, some earlier about the 15th, there's a high tunnel workshop. The 16th, we'll have a cover crop workshop. And then I know Liz Robinson has been posting about her Urban Farm Fest, which is April 21st. So yeah, I guess, watch out for those in the Google group, and we'll be opening an Eventbrite registration up for the cover crop workshop pretty soon. And Terry, I don't know if you have any announcements. My colleague Terry.
No, I'm good.
We do have the cooperator dinner coming up on, when is it, Terry? April 19th?
February. Oh my goodness. Friday, April 19th.
I still need to send the invite out to the Google group, but I'll be sharing that shortly.
Okay.
And I think that's it. So thank you again. Lisa, I'm going to go ahead and stop the recording. If anyone wants to hang out for a few minutes, we'll hang on if folks have individual questions. But otherwise, we'll see you next month. And happy equinox, everyone.
Yeah. Good night, everyone.